In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum. How are you all? I once again welcome you in Pakistan International School Types virtual learning class for second year biology. Dear students, we have started chapter number 16, support and locomotion. We have seen all about bones, its distribution in the body, joints, disorders of the skeleton. Today we will see about muscles. Our topic is muscles. We will see the structure and functions of different types of muscles, its mechanism of contraction, as well as we will see some disorders, inshallah. So let's get started. Dear students, muscles. We know that muscles are specialized tissues which undergo contraction and relaxation, and it provides movements to every part of the body. So the study of muscles is called myology. Myo means muscles. Ology means its study. So it gives and it performs the functions for the positions, for the movements of the body parts, as well as certain fluids in the body, like smooth muscles can do perform their functions in the body. So we will see here types of muscles. Types of muscles are of three: smooth, cardiac, and skeletal muscles. So the names are describing you the types. What are smooth muscles, what are cardiac and skeletal muscles? We will see one by one. Smooth muscles are actually called smooth because it does not have striation in between, or they are having, you can see here in this diagram, that smooth muscles are having like this a spindle formed structure like this, and they are having one nucleus, large nucleus in between, and like in the center of the cell. It is a spindle like a structure. It does not have a striation, means these type of bands, dark and light bands, which are present in cardiac or skeletal muscles. You can see it's such type of lines. These are called striations. So they are not present in the smooth muscles, and they are joined together with their spindle formed from here with one another, as well as myofilaments are not organized in sarcomeres. Like what are sarcomeres? We will see in detail in skeletal muscles. Sarcomeres, actually, that is the unit of contraction of the muscles. So they are not well organized as in skeletal muscles are. That's why striations are not appeared. They are jumbled like in smooth muscles. That's why they appear smooth, not striated. And they do not have intermediate filaments as well, which join both of the, like this is one spindle a type of the cell, and the other will join from here like this. So they do not have any intermediate filament which join both of the cells together. That's why they do not have striation as well. They are present in every internal organs, like blood vessels are there, which provide like easy flow of the blood, urinary tract as well, reproductive system and digestive system, as well as respiratory system like our windpipe, and in other places also. If we see cardiac muscles are somehow related to the skeletal muscles, but they are the specific and the strongest muscles of the body. They are specific to the heart only, and they are branched. If we just see in this diagram, these are like they are just somehow similar with the skeletal muscles, but we can say they are as similar as the skeletal muscles. Okay, as well as they are having two cells attachment, where the cell to cell attachment is there, that is called intercalated discs, and that which is having a gap junction, which allow action potential to pass from cell to cell, like in whole of the heart. If we see here skeletal muscles, so they are attached to the bones and are responsible for the movement of other body parts. So we can see here also the types of muscles. If you see the heart muscles, these are the heart muscles or cardiac muscles. They are having branch structure, and where they join together, they are having intercalated discs, which is having junction like the space, some space or junction by which the action potential generated in the heart muscle, it can be spread easily. These are skeletal muscles, like they are parallel to one another, and these are, we can see, the spindle shaped smooth muscles. They don't have striations, and they are present in the soft tissues or soft parts of the body. So if we see here, these are actually, that is I think incorrect writing here, that is the mistake. There is cardiac muscles, and these are skeletal muscles, because the intercalated discs are present like this when two cells are joined, this one and this one are joined together. So they are present in cardiac muscles, not in skeletal muscles. So we can see here the differences between three type of muscles. If we see its appearance or structure, they are unstriped or striped, and the cardiac and skeletal muscles are having striped, where cardiac muscles are having irregular striped, and skeletal muscles are regular striped muscles. So if we talk about cell shape, so smooth muscles are spindle shaped, cardiac muscles having branching system, and skeletal muscles are cylindrical or sometimes spindle shaped as well, like where they are attached with the bones, so they are having tendons. So with the tendons, they form spindle shaped structure. Number of nuclei, so in smooth muscles only one per cell is there, one nucleus per cell. In cardiac muscles also one nucleus per cell, but in skeletal muscles there are many nuclei per cell. Speed of contraction in smooth muscles is slow, and cardiac muscles intermediate, and skeletal muscles slow to rapid. When there is a rapid movement, so there is increased speed, and if sometimes there is very slow when we do function very slowly. Contraction used by nervous system in smooth muscles as well as in skeletal muscles. But if we talk about cardiac muscles, so cardiac muscles are spontaneous, like nervous system is not involved in this one, but heart muscles generate the action potential within by itself within the heart with the pacemaker. If you have the name, because you have studied this in grade 11. So functions control movement of substances in hollow organs, like in the digestive system, they control the movement, as well as our cardiac muscles pumps the blood, and the move skeleton is done by skeletal muscles. Voluntary control, so usually there is what we can say, no control of smooth muscles because they are involuntary, I think so. And usually no control over there, but skeletal muscles are having a control, or we are having control on them. Okay, let's talk about the structure of skeletal muscles. So skeletal muscles are actually, yes, they are striated muscles as we have seen before, and there are alternate light and dark bands as well, and which we can see under the microscope only. And they are composed of muscle fibers, or those are called muscle cells. Each skeletal muscle fiber is single cylindrical cell which is enclosed by plasma membrane. Yes, we will talk about in detail. So first we will see here in the structure, you can see here, uh, this is the skeletal muscles, biceps and triceps of the forearm. So you can see here the structure of muscles externally. So these are joined with the bones, with the tendons, and then we can see these muscles when in electron microscope, enlarged view of that muscle. So you can see here that one muscle fiber, if we take it this muscle structure, closer look of that, so it is covered by the outer covering that is called epimysium. So if we see muscle bundle, and in that if we take one fiber from that, that is called fascicle, and this fascicle is covered by a connective tissue that is called perimysium. And now if we take this fascicle out, if we take this fascicle out, so we can see here there are also muscle fibers, lots of muscle fibers in them, and these muscle fibers are also called muscle cells. So these, each muscle fiber is covered by another protective layer of connective tissue that is called endomysium. Endo means internal, so that is very internal or inner layer which covers this one. When we take this muscle fiber out, that is muscle fibril, and that is having dark and light bands in it, and these light and dark bands in it, and these light and dark bands are composed of very specific proteins. Those are called actin and myosin, and these are called actin myosin filaments. If we just take a look on the brief structure, like on this structure in detail, so you can see here that this is we can say the piece, little piece of that band, and that is called muscle fiber or myofibril, and this is having dark and light bands. So they are composed of, you can see here, certain lines which are darker, and here some are lighter here. And now we can see this in detail. So you can see here, this is the central line. This central line is called M line. M line is here and M line is here. And that is the connection of the, or we can say that connects myosin fibril of right side with the left side. Like you can see this one here. And this is actually in the center of two myosin filaments. And this is called myomesin because it is made from myomesin protein. Okay, these dark lines which you can see here, these are called myosin filaments, and the lighter one, this blue, these are called actin filaments. You can see one more line, that zigzag line. These lines join actin filament of one side with the other side. So from one Z line to the other Z line, this portion is called sarcomere. And this sarcomere is actually from here to here. Actually, this is the unit of what we can say, the unit of a contraction. So the sarcomere is the contractile unit of the muscle contraction. Okay, you can see here there are some bands like A band, I band, then this is H zone, M line. These are all indicates the distribution of like different portions of this contractile unit. So if we talk about this Z line, so this Z line, I have mentioned this before, that this is the connection between two actin proteins, or that is the lighter band. This is actually we can say it Zwicken Skebe. Zwicken Skebe, that was like actually what its meaning is between discs, and this is the German word. So between two discs, like from one Z line to other Z line, there is one sarcomere. So from here to other Z line, there is other sarcomere. So between two sarcomeres, two discs, there is a Z line. If we talk about this A band, so if we see this one, you can see this in detail here. Below, this is A band. A band is the portion of that sarcomere in which just only this a thick region or thick filament is present, that is called myosin filament, and this is present at the A band. So because this is the ultrastructure of sarcomere, and this is anisotropic band, it means it can polarize the visible light. That's why its name is A band. You can see here from here to the other region of this myosin filament, which is here. Now, if we see this, this is H zone. H zone is the portion where only we can see myosin filaments are present, just darker region, not overlapping of this actin filament is there. So that is called H zone. That is stands for hele. Hele means bright. So here actually not overlapping of these two. That's why it looks brighter in color. So this is M line. M line we have discussed before. That is like the connection where two myosin filaments are connected with one another, and this is the common point of both of the myosin filaments. Actually, when the contraction is done, what happened? Just these thin filament, that is actin filaments, they move inward, and by the help of myosin filaments, and myosin filaments just remain at their places. Now we will just talk about after a few minutes about that. So you can see here, like if we see the cross section of this sarcomere, so we can see that these, the darker, the red one are myosin filaments, and the blue one are actin filaments. So one actin filament, this one is like is covered by 3 myosin filaments, and one myosin filament is covered by 1, 2, 3, 4, 5, 6 actin filaments. Okay, if we see here the ultra and deep structure of myosin and actin filaments, so you can see here in this diagram, or we can move to the next word onward. You can see just a first brief of this one. This is bone, tendon, a skeletal muscle, then perimysium is this one, the upper one is epimysium, and now it is having fasciculi, this plural, and in which we are having muscle fibers, and in muscle fibers covered by endomysium, and then we are having muscle fibers out one muscle fiber, so that is called muscle fibril. And look, you can see on that lots of nuclei are there, capillaries are over there to provide blood supply, as well as there are like sarcoplasmic reticulum, because this is very, very important organelle of this muscle fiber. What it does, it provides like uh, flashes of calcium in the muscle fibers. Due to that, the muscle fiber contracts. We'll talk about in its functioning. So this is T tubule or transverse tubule that is actually along all the muscle fibril, and like you can see here, that is along all the muscle fibril, and that is to provide action potential which comes from the nerve, any type of nerve cell into the muscle fiber. Now we will have muscle fibril, and you can see sarcolemma is there. Sarcolemma is actually the outer covering of the muscle fibril, and like muscle fibril is one cell of like we are having our body cells, so our body cells called plasma membrane or cell membrane, so this is called sarcolemma. Sarcolemma is also the plasma membrane of this muscle fibril. Then we can see here lots of mitochondria are present here, which provides ATP, which is the chemical energy which changes into mechanical energy, and which provides like the functioning or contraction, which do the process of contraction of muscles. If we take the fibril out, then we can see striation. These striations are due to the H band, H zone, and Z, like the dark band, A band, and I band. And that is due to the overlapping of actin myofilament and myosin myofilament. Myo means muscle, so that is the filament of muscle. Now we can see the ultrastructure of actin and myosin, and we can see its structure. Then we'll talk about its functioning as well. So you can see here the Z disc or Z line is present, this blue one, and from here to here, that is the ultrastructure of that sarcomere. This is sarcomere, in which there is M line by which these myosin filaments are connected of both of the sides. Here is myosin filament and actin filament. These are bead like structures, And these beaded like structures are made up of certain proteins we'll discuss after some moments. Then we are having their cross bridges. Cross bridges means this myosin filament makes the cross bridge with actin filament, by which it just helps in the contraction of the muscles. Then we can see here titin proteins, and these proteins are that makes the filament, spring-like filament actually, that gives the elasticity to the muscle, as well as it keeps the myosin filament in its position. Okay, so if we now we have seen like I band, that is the lighter one. H zone, in which just myosin filaments are there. A band, in which actin and myosin filaments both are present here, as well as Z line, M line, and we have seen some other proteins like titans. Okay, so if we just see, we have seen actually ultrastructure, but if we just talk about the structure of actin and myosin filaments, so you can see here that myosin molecule consists of uh, six polypeptide means chains, polypeptide chains, which are arranged in such a way that each myosin molecule possesses a tail and two globular heads. So they are just twisted together, and they form a tail, and they form two heads and one tail. So each thick filament contains about 300 myosin molecules. Like this is one, and then you can see here one, this one, then two, then three, then four, like this. So there are almost 300, and all make one long tail by twisting around one another. And they're facing heads, their heads are facing outward and in opposite direction to at each end. Like here is the other one, and here is this one. So they do like this, they're facing other direction. So if we just talk about actin protein, and actin protein is kidney-shaped polypeptide subunits of actin are present over there, and they are made up of globular actin, or globular means G actin protein, and they bear the active sites to which myosin heads attach during contraction. G-actin monomers, like this is one monomer. This is almost kidney like that. It's round from here, but it is having some structure like this from this side, because here it is having the connection with myosin filament. So we can see that when these G proteins just bind together to make pearl or beaded strand-like structure, that is called fibrous or F-actin. So the backbone of this each thin filament appears to be formed by two intertwisted, you can see it, two intertwisted F-actins. And if we just see this actin protein, so that is 3 to 8 nm thick. And if we talk about myosin filament, so that is 16 nm thick, and that is made of myosin protein, and actin is made up of actin protein specifically. But with actin protein, there are some other proteins are present. Two strands of tropomyosin spiral around each actin, uh, like um, polypeptide. You can see here this strand, this rope-like structure. This is the protein that is called tropomyosin, which is twisted around it, and this helps to bind the sides this type, these are the sites from where it binds with that myosin filament. So this tropomyosin, just when it is in relaxed position, so this is covered, or this is bind with tropomyosin, so that this site is not available for contraction right now. So troponin is actually a 3 polypeptide complex is there. If you see here, this is 3 polypeptides, 3 types of troponin proteins are there. So one is TNI. TNI is actually I for inhibitory, TN for troponin. So this is inhibitory subunit that binds to the actin, and from here, these are 3. So one binds, that is present on tropomyosin, so that binds with this with actin, so that it covers that binding site which will be available at the time of contraction to myosin filament. That's why it is called inhibitory subunit. The other one is TNT. 3NT binds to tropomyosin and helps position it on the actin. This helps this tropomyosin, and it binds and it keeps this this actin filament in its position. And the third one, that is TNC. TNC, this is the subunit which binds with calcium. So when there is like muscle contraction, so what happened, this is the calcium, this is the subunit which binds with calcium, and the contraction starts. So both a troponin and tropomyosin help control the myosin actin interactions involved in contractions. So here you can see TNI, TNT, and TNC. This is troponin, calcium, which binds with calcium, troponin, which binds with tropomyosin, and troponin, which is inhibitory subunit that binds to actin. Okay, dear students, that's it for today. Inshallah, we will continue our the next topic, muscle contraction, in the next lecture. Wish you all the best. Allah Hafiz.